Okay, so good day again. So as what I've told you, today I would be discussing chlamydia. So let us discuss first chlamydia. So chlamydia is known as obligate intracellular parasite. Okay, so when you say obligate intracellular parasite, they cannot live outside the host cells. Okay, so particularly for chlamydia, it's quite difficult for us to cultivate them uh, using artificial culture media. So they are difficult to culture. So in order for us to culture them, we need live tissue culture or live tissue or cell culture. So noteworthy to mention, particularly for chlamydia, we are using the cell line known as the McCoy cells. So we are using McCoy cells. So molecular biology assays affect in the organism's identification. So members of this particular genera include Chlamydophila, Rickettsia, Orientia, Anaplasma, and Ehrlichia. Okay, so for today, we will be focusing first on Chlamydophila. So let's compare them. How do we compare this particular group of organisms with typical bacteria and with viruses? Okay, and also with mycoplasma. So mycoplasma is an example of bacteria. It is a bacteria without peptidoglycan, meaning to say this group of bacteria is acellular because they do not have cell wall. Viruses are not bacteria, so they are filterable agents. They are smaller than bacteria and they cannot live outside the host cell. Hence, they are termed as obligate intracellular parasite. Now, typical bacteria would have peptidoglycan. They have peptidoglycan and they have cell wall. Therefore, since they have cell wall, we can identify them as gram-positive or gram-negative. For mycoplasma, they do not have cell wall, so we cannot identify them as gram-positive or gram-negative. That's not possible because they do not have cell wall. So where do Chlamydia and Rickettsia will fall? Okay, so take a look at the composition, DNA and RNA. All of them are bacteria. So tandaan ninyo ha, from this up to these are all bacteria. And bacteria will possess both nucleic acid, meaning to say they have both DNA and RNA. But for viruses, they can only have either DNA or RNA. A virus cannot possess both nucleic acid. So for example, the SARS-CoV-2, which is the etiologic agent of COVID-19, is an example of RNA virus. Herpes virus is full of DNA virus. So a virus cannot possess DNA and RNA. It can only possess either DNA or RNA. So that's why it says negative here. But for the rest, these are all bacteria, meaning to say they can possess both DNA and RNA. Obligate intracellular parasite. So chlamydia, rickettsia, and viruses are considered to be as obligate intracellular parasite, meaning to say you cannot cultivate them in artificial culture media. You'll be using you will be using live tissue culture. How about the presence of peptidoglycan in the cell wall? So a typical bacteria would have peptidoglycan, the same with chlamydia. However, Rickettsia, Mycoplasma, and viruses do not have peptidoglycan. How about growth on non-living medium or artificial medium? So typical bacteria can grow on artificial culture media, and so with Mycoplasma. However, chlamydia, rickettsia, and viruses cannot grow on a non-living medium. They can only grow in live tissue culture. How about ribosomes? Ribosomes are very important for protein synthesis. So all of them has ribosomes except for viruses. Viruses, it says here it's negative because viruses would need to use the genetic machinery of the host in order for them to replicate. So they don't have their own ribosomes. How about sensitivity to antimicrobial agents or sensitivity to um, anti antibacterial agents? Let's be more specific. So typical bacteria, chlamydia, rickettsia, and mycoplasma, all of them are bacteria. Therefore, they are susceptible to antibiotics. Viruses are not bacteria, so you cannot give antibacterial agents to treat viral infections. How about sensitivity to interferon? So, what is an interferon? Interferon is a protein and it has an antiviral property. Okay, so therefore, viruses are susceptible to or sensitive to interferon. However, they found out that chlamydia, although they are bacteria, they are also sensitive to interferon. How about binary fission? Binary fission is actually the mode of replication for prokaryotes. And since all of them are bacteria, all of them are prokaryotes. Therefore, they are able to do binary fission, except for viruses. So viruses replicate by means of protein synthesis using the genetic machinery of the host. Okay, so that's how we differentiate um, bacteria, typical bacteria, Chlamydia, Rickettsia, Mycoplasmas, and viruses. So for this particular module, I will be focusing on two important organisms, the Chlamydia and the Rickettsia. Okay, so moving forward, let's discuss the first subtopic in this module. So we will be discussing Chlamydia C. Okay, so do you know that chlamydia are tiny intracellular bacteria and they are considered to be as energy parasite. So chlamydia are considered to be as energy parasite because they obtain energy for growth from their host cells. So they synthesize the ATP coming from the host cells. So that's the reason why they are known as energy parasites. Okay, so chlamydia are obligate intracellular parasites of humans that can cause trachomatis and pneumonia and even infect cetacean birds or parrots. Okay, so this uh, the term chlamydia is actually from the Greek word chlamylo, which means cloak. Okay, the Greek word cloak, because it is as if this is the nucleus, okay, it is as if something is surrounding around the nucleus. So later on, we will be discussing that there are two important parts of the chlamydia cells. So we will be discussing that we have elementary body And then we will also have the reticulate body. So these are important components of a chlamydial cell, hence from the term cloak, chlamylo, which means cloak. Okay, so but before we discuss that further, let us differentiate first the different chlamydiaceae species. So there are three medically important species of the chlamydiaceae family. Okay, so we have the chlamydia trachomatis. Okay, we have the chlamydophila pneumoniae, and then the chlamydophila psittaci. Okay, so the inclusion bodies for chlamydia trachomatis is round and bacular. Okay, for chlamydophila, they are round and dense. For Chlamydophila psittaci, they are variable shape and dense. So when you say vacuolar, it is as if it is clear and with halo-like structure. Okay. So glycogen is part of the inclusion bodies for Chlamydia trachomatis, but not for other species. The elementary body, okay, which is again a part of the chlamydial cell, is round for trachomatis, but they are pear-shaped for Chlamydophila pneumoniae and also round for Chlamydia psittaci. And then sulfa drug sensitivity. So we can actually use sulfa drug for the treatment of Chlamydia trachomatis, but not for Chlamydophila pneumoniae and Chlamydophila psittaci. DNA relatedness against Chlamydia pneumoniae. Okay? So, against Chlamydophila pneumoniae. So, surprisingly, both Chlamydia trachomatis and Chlamydia psittaci are only 10% related to Chlamydophila pneumoniae in terms of DNA similarity. Okay? And for the natural host, of course, Chlamydia trachomatis and Chlamydophila pneumoniae, its natural hosts are humans. And for Chlamydia psittaci, the natural hosts are birds, birds and lower animals. So, noteworthy to mention also that for Chlamydia trachomatis, it causes one of the most common yet not popular sexually transmitted disease. So we call it chlamydia. Okay, so this particular sexually transmitted disease is known as the LGV. So later on, we will be discussing that. Okay, and lymphogranuloma venereum, trachoma. Okay, and Chlamydophila pneumoniae may cause pneumonia, pharyngitis, and okay, and then Chlamydophila psittaci may infect bird, lower animals, may also cause pneumonia. So clinical infections, as what you can see here, Chlamydia pneumoniae causes pneumonia and human-to-human infection is possible. Chlamydia trachomatis may cause sexually transmitted disease. Human-to-human infection is possible. Chlamydia psittaci, its natural host is a cetacean bird, and bird-to-human infection is possible. So they are energy parasite because they are deficient in energy metabolism, resulting for being an obligate intracellular parasite. So again, there are two important parts. So we have the elementary body, which is the infectious particle or the infectious part, okay? And then the major outer membrane proteins. The MOMP is important antigenic property of the bacteria. Hence, since it is important for antigenicity of the Chlamydiaceae, the MOMP is being detected by monoclonal antibodies. And then the other part is the reticulate body. So the reticulate body is the non-infectious part of the Chlamydiaceae. So chlamydia are intracellular parasites similar to gram-negative bacteria, but they lack peptidoglycan. between the inner and outer membrane. So if, for example, if this is the inner membrane and then this is the outer membrane, so peptidoglycan is supposed to be here. So their life cycle is unique because in the life cycle of chlamydia C, you would notice that there are two important parts, okay? The elementary body and then the reticulate body. So what is the elementary body? So the elementary body is the infectious agent, meaning to say the elementary body can invade host cells. So this is the host cell, and then this is the nucleus. So the elementary body is the infectious particle, and they are metabolically inert or active. And this one is being released by the infected host cell. So if you will be looking at the morphology of the elementary body, they have a very rigid surface. Okay, they have rigid surface proteins with many disulfide links. So the disulfide links would allow the elementary body to survive outside the host cell. So the rigid surface protein takes place of the peptidoglycan layer. So ordinary bacteria would have peptidoglycan in their cell wall, but for chlamydia, instead of peptidoglycan, they have the rigid surface protein. Now, the reticulate body is being phagocytized into columnar epithelial cells. So for example, if we are talking about blindness, it could be columnar epithelial cells in the eye, or if we're talking about sexually transmitted infection, these are the epithelial cells in the genitalia. Or if we're talking about pneumonia, we're talking about epithelial cells found in the respiratory lining, for example. Okay? And then, once inside the cell, the elementary body is reorganized into a reticulate body that is metabolically active. So let me correct myself. So the elementary body is metabolically inert. So when you say inert, they are inactive. Okay? But once inside the host cell, elementary body is reorganized into reticulate body. And they are now metabolic active. Okay? So the reticulate body divides by means of binary fission. And the large number of reticulate bodies produced will form an inclusion in the cytoplasm of the cell that rings the nucleus. Hence the term clamilo, which means glow. So if I will go back to the slide, to the previous slide, this is the nucleus, and then this is the reticulate body. And the reticulate body is actually um, formed around the nucleus. Hence the term clamilo. Okay, so that's how chlamydia derive its name. Okay, so chlamydia inclusion surrounding the nucleus. Okay, so I hope that's clear. So here's another one. So this is the nucleus, and then this is the inclusion, and this is the entire cell. Okay, so that's another example. So the kalamilos is a Greek word for cloak, which means around the nucleus. Okay, so again, this is the nucleus, then this is the inclusions. Okay, and 18 to 24 hours following the infection, the inclusion contains both the elementary body and the reticulate body forms. So within 48 to 72 hours, the cell will rupture and it will release several elementary bodies. Okay, so these particular elementary bodies will now infect another host cell. So chlamydia cannot synthesize ATP and therefore considered to be as the energy parasite. So it will infect another host cell. So that is the life cycle of chlamydia. Okay? So I hope that's clear. So two important parts, the elementary body and then the reticulate body. Okay, so that's the another example of life cycle in a nutshell, no? So again, so elementary body will phagocytize, it will be reorganized into reticulate body, they will divide, multiply, and then they will now start to release the elementary body. So again, that's another example of the life cycle. Okay, so let's dwell more on reticulate and elementary bodies. Okay, so here are examples of elementary bodies seen outside the host cells. And then these elementary bodies are highly infectious. Okay, and they can even infect another cell. Okay, so let's discuss now Chlamydia trachomatis. So there are two biovarieties of Chlamydia trachomatis. Okay, these biovarieties are the trachoma and the other one is the lymphogranuloma venereum. Okay, characterization of the MOMP or the major outer membrane protein has actually separated Chlamydia trachomatis into 20 serovariants or serovar. So again, it's actually possible because the MOMP is important for the antigenic properties of the chlamydia. So trachoma biobar include serobars of A to K. So imagine there are A, B, C, D, E, F, G, H, I, J, K. So that's already an 11 biovariant. Okay? So why is it important to determine the serovarieties to that of the biovarieties? So again, there are two biovarieties. Biovarieties because this particular Chlamydia trachomatis can cause two major illnesses or infections. So the first one is trachoma, and the other one is the lymphogranuloma venereum. Now, why is it important to determine the serovarieties? Because the serovarieties will somehow be responsible to what type of infection it can actually give. So, for example, serovarieties A, B, B, A, and C are responsible for the severe eye infection known as the trachoma. And do you know that trachoma is the world's leading cause of blindness? Okay? Serovarieties D to K, DA, IA, JA is important etiologic agents of inclusion conjunctivitis, milder eye infection, and urogenital infection. Okay? And then, aside from that, we also have lymphogranuloma venereum serovirus. So, this may be known as the LGV1, 2, and 3. They are associated with invasive urogenital tract disease. Okay? Lymphogranuloma venereum. Okay, so these are the different serovarieties and biovarieties. Remember that there are only two biovarieties, but there are about 20 serovarieties, particularly of Chlamydia trachomatis. So let's talk about the eye infection. So the eye infection is known as the trachoma. It is a chronic eye infection because a long-term infection with this bacteria will lead to blindness. But The good thing about this blindness is that this one is preventable. So they are frequently seen in the equator, wherein, syempre, if you're near the equator, so the place is high in temperature, high in humidity. So it's not common in temperate countries such as in the United States. I'll just plug in my charger. So they can produce chronic infection that, if left untreated, will generally end in blindness among the adults. So there are actually four stages of trachoma. So the first stage is asymptomatic with little conjunctival exudate. So exudate is actually a fluid that is produced due to inflammatory process. So exudate is high in protein, high in specific gravity, and it has many WBCs or PAS. Think you are discussing this in AUBF. And because of the inflammation, there is a follicular and papillary hypertrophy. I think papillary is supposed to be spelled this way. Okay? And then because of that, long-term infection. So this is actually an acute inflammation. Stage number 2 is an acute inflammation as well as stage number 1. But this is now the chronic effect. Scarring of conjunctiva and panus. Okay, scarring will result if it is untreated. But if you will be treating it, trachoma could be healed with no evidence of inflammation. So that's the reason why blindness is actually preventable. Okay? So repeated infection is often spread by flies from one child's eye to another. And this can actually gradually lead to panus and scarring of the cornea. And if your cornea is scarred, every time you every it the can actually scratch the cornea. Okay, the scarring of the panus can actually scratch the cornea. And that is the reason why it can lead to blindness because of the chronic untreated trachoma. Okay? So, trachoma is different from inclusion conjunctivitis. Although, inclusion conjunctivitis is also an eye infection. So, usually, um, it is it happens when a newborn passes through the birth canal and the mother is infected with Chlamydia trachomatis serovariety D2K. And it results in conjunctivitis 5 to 12 days after birth. And the infection may persist for months if not treated. So you usually see here, there is a pus formation, which indicates that indeed there is an inflammation. Okay? So all newborns, therefore, should be treated with erythromycin eye drop. This will prevent infection with Neisseria gonorrhoeae. So if you remember our lesson, Neisseria gonorrhoeae causes ophthalmia neonatorum. So fortunately, both Neisseria gonorrhoeae and chlamydia are inhibited by erythromycin. So it's very important that we use an erythromycin. Actually, it's already a law that all newborn should receive erythromycin drops at birth. In adults, the organism is acquired either through sexual contact or self-inoculation. So if someone touches his infected or her infected genitalia and unconsciously touches the eye, so that's how inclusion conjunctivitis would result. It would actually result to complete inclusion conjunctivitis. Okay, so what are the virulence factors? Okay, so attachment, because the organisms have the ability to attach to target cells. The target cells are the non-ciliated columnar epithelial cells, and these are found in the mucous membranes of the conjunctiva. So in our eyes, urethra, cervix, endometrium, fallopian tubes, and rectum. So these are the genitalia, and even in the respiratory tract. Hence, it can also cause pneumonia, some species, the other species. So the one that can attach to the respiratory tract epithelial linings are Chlamydophila pneumoniae and Chlamydophila psittaci. Okay, so Chlamydia trachomatis therefore has several biovarieties. So A, B, BA, then C may cause trachoma. And then serovarieties D, DA, E, F, G, H, I, A, J, A, K. They can cause inclusion conjunctivitis, non-gonococcal urethritis, cervicitis, salpingitis. Okay, dito sa salpingitis, the reproductive organ of the female is already affected. Pelvic inflammatory disease, endometritis, acute urethral syndrome, proctitis, ito naman sa male, epididymitis, pneumonia of newborn, and perihepatitis, or the Fitzhugh-Curtis syndrome. Okay? Ito, both adult and newborn. Now, let me emphasize to you NGU. NGU stands for non-gonococcal urethritis. Okay, non-gonococcal urethritis, which means that persons would have symptoms similar to gonorrhea. So if someone has NGU or non-gonococcal urethritis, you would expect him to have symptoms similar to gonorrhea, except that it is negative for gram-negative diplococci. That is actually Neisseria gonorrhoeae, and it is non-cultivable. Okay, not cultivable in non-living culture media. So aside from chlamydia, okay, ureaplasma may also be an example of NGU or non-gonococcal urethritis. So again, a person with NGU would have symptoms, signs and symptoms that are similar to gonorrhea, except that it is not caused by gonorrhea. It could be caused by chlamydia, trachomatis, or ureaplasma, urolithicum. And another important species that we have here is Chlamydophila pneumoniae, and there's only one serovariety. So this would cause pneumonia, bronchitis, pharyngitis, and the influenza-like febrile illness. So Chlamydia trachomatis and Chlamydia pneumoniae affects humans. And the one that would have birds as a natural reservoir is Chlamydia psittaci, and there are about 10 serovarieties. So this can cause psittacosis, endocarditis, and abortion. Okay, so that's okay. In a nutshell, these are the different diseases, human diseases caused by Chlamydia species. And so this is an example of conjunctival scarring and hyperendemic blindness, okay, caused by untreated trachoma. Okay, so take note that the reason why it is red because of the papillary hypertrophy. So let's talk about LGV. So LGV is lymphogranuloma venereum. Okay. Um, the serovarieties are the L1, L2, L2A, L2B, and L2. So it is also linked to the parotid oculoglandular conjunctivitis, the one that you can see here is an example of the parotid oculoglandular conjunctivitis. So here, the organisms will survive in mononuclear cells. So there would be bubo formation. Okay? So bubo is actually the swollen lymph nodes. So the lymph nodes will be swollen and painful, and it is actually a uh very once it is swollen, then eventually it will rupture. Okay, so it is highly prevalent, though we do not have exact records of its prevalence in our country, and as it is found in tropics and subtropical areas. So our country is a tropical area. So yes, it's common in our country. So the primary infection is usually from an organism gaining entrance through minor abrasions or lacerations. So as you can see, there is a swollen lymph node here. Okay, so lymph nodes near the primary focus swell with inflammatory cell to cause a painful swelling or the bubo. Okay, so this is another swollen lymph node in female. So you can see here the swollen lymph node. Okay, so incubation period is between 1 to 4 weeks, and the patients who develop a small papule or lesion at the infection site, so one that you can see here, followed by swelling of inguinal, okay, lymph nodes, not regional, sorry, inguinal lymph node. So the causative serovarieties are able to survive inside the mononuclear cell. So bubo formation may occur after bacteria will enter the lymph nodes, producing strong inflammatory response and subsequent rupture of the lymph node. Okay, so it is common in women from lymphatic spread of the bacteria from the vagina or cervix. It is also possible in men, particularly those who are involved in anal receptive intercourse of the lymphatic spread from the urethra. Okay, so here is an example of an infected anus. And this is what you call the proctitis. Okay, so see here, the dentate line is actually this one. So because of the receptive anal intercourse. So here is another example of inguinal swelling and lymphatic drainage, okay, of a chlamydia infection. It may also cause urogenital diseases in women. So, for example, an inflamed um, fallopian tube or the salpingitis, cervicitis, endometritis, and proctitis. So, if a woman is infected with chlamydia, long-term infection may result into pelvic inflammatory disease in which she'll find it difficult to get pregnant, or the chances of getting ectopic pregnancy or pregnancy outside the fallopian tube would be highly possible. Perihepatitis, the another complication similar to gonorrhea, is also a possibility. So remember, perihepatitis is also known as the Fitzhugh-Curtis syndrome. Asymptomatic infections are common, though they are asymptomatic, they are still infectious. Okay. There are also other urogenital diseases in men. So again, They can cause NGU, and possible complications include epididymitis, prostatitis, and proctitis. So I have already explained what proctitis is. So again, for male, asymptomatic infections are also common. So here is an illustration of an inflamed epididymis and testis. Because of the long-term infection, Reiter syndrome will occur. So Reiter syndrome is characterized by reactive arthritis, and it is believed to be caused by Chlamydia trachomatis. So Reiter syndrome is characterized by urethritis, so that is the inflammation of the urethra, conjunctivitis, polyarthritis, and mucocutaneous lesion, such as the one that you can see here in the photograph. So, Chlamydia trachomatis is the most common sexually transmitted bacterial pathogens in the United States, although I do not have data here in the country. Many infections go undiagnosed, probably because of the presence of asymptomatic carrier state. So, only genital wart is different from chlamydia because genital wart is caused by a viral infection known as the human papilloma virus. So, human papilloma virus, though, is more common in the United States as compared to the chlamydia trachomatis. Chlamydia infection in newborn is also a common notable manifestations. So, chlamydia infection in newborn is characterized by conjunctivitis, nasopharyngeal infections, and pneumonia. So, aside from that, it is also possible that otitis media is present. So it's actually preventable, as what I mentioned to you. We're using erythromycin, as this will prevent not only chlamydia trachomatis, but it will also prevent Neisseria gonorrhoeae as well. Okay, so inclusion conjunctivitis in neonate would have 4 to 5 days incubation period, and you would notice that the eyelid is swollen, so it becomes edematous, and you'll see that the secretions here, okay, the discharge here is copious and yellow, so these are actually pus. If untreated, it will last for about weeks to several months, and complications will lead to corneal panus formation and conjunctival scarring, and this may result to blindness if untreated. Okay. So, what are the laboratory diagnosis consideration factors? Um, Before the clinicians would warrant the testing of chlamydia, so a clinician would need to assess the following. So, the knowledge of population at risk. So, he should have the clinical history. And then, of course, very important also is the capability and facilities available for testing. Okay? Ability to batch specimen types, because especially if the clinic is far, so they should be able to store all of the samples and send it all at the same time. And of course, the experience of the laboratory scientists. So the assays may be divided into two, culture and into four actually. So it could be culture, non-culture, non-amplified, and then culture amplified. When we say culture amplified, here we're talking about PCR. And then serological test. Okay? So for prenatal, um, it is useful. For newborn, eye is probable, but throat is also useful. For clinics, um, for low risk, it is most useful. Okay. Legal applicability, yes, it's legal. Okay. Test of cure, it's also possible. Okay. But for non-culture, non-amplified, meaning to say, we we detect it by means of direct fluorescent assay, enzyme immunoassay, and then OIA, sorry, OIA, is another type of immunoassay, but they didn't indicate. Oh, maybe it refers to the outer. I'm not sure because lipopolysaccharide, outer membrane protein immunoassay. So it's not really clear what OIA is here. So maybe I can actually check Mahon later on. For non-culture, so we can use PCR. SDA is the strand displacement amplification, and then TMA is the transcript mediated amplification. For serology, this one is the most practical serological test. We can use complement fixation, enzyme immunoassay, and MIF. OIA is optical immunoassay. Nandito pala guys. OIA is optical immunoassay. And MIF is microimmunofluorescence assay. Okay, so let's see how useful they are one by one. Okay, so when submitting the specimen in the laboratory, usually if you want to culture them, we need to collect swab. And we usually swab the lesion using the dacron, the rayon, or calcium alginate swab. So we do not use wooden shaft. Wooden shaft are toxic, so we either use plastic or metal. So we need scraping. Discharge is not enough. For gonorrhea, discharge is enough. But for chlamydia, we need scraping. And for urine and vaginal swab, first morning urine and vaginal swab are excellent specimen. Okay? Specimen for culture must be placed into a transport medium and transported at 4 degrees or lower within a day. So within 24 hours, you need to send it to the laboratory. Okay, so these are the specimen. Okay, so for inclusion conjunctivitis, we need the conjunctival swab. For urethritis, we need a urethral swab. For epididymitis, we need an epididymis aspirate. For cervicitis, we need an endocervical swab. For salpingitis, we need a fallopian tube or biopsy. Okay? If we need to clarify STD, then we actually use rectal or vaginal swab. For sexually transmitted disease for of the male sex partner, then urine is also being collected. For lymphogranuloma venereum, which means that there is already a swollen lymph node, then we collect bubo or the cervical lymph node aspirate. For infants pneumonia, we do the swab throat or the nasopharyngeal aspirate or the lung tissue. For chlamydophila pneumonia, we do throat washing, throat swab, sputum, bronchial lavage, fluid, or even the lung tissue. For psittacosis, we collect sputum, bronchial lavage, pleural fluid, and also lung tissue. Okay, so these are the possible specimen needed for the detection of chlamydial infections. Okay, so let's discuss first direct microscopic examination. Okay, when you say direct microscopic examination, we stain and examine directly the specimen. Okay, we stain and directly examine the specimen, meaning to say we do not we do not examine the specimen from the culture, but we directly examine the specimen. So we can either do cytologic methods, and the other one is the direct fluorescent antibody testing. Okay. So the cytologic method is 95% sensitive, but it's technically demanding. So it is influenced by specimen quality and experience of the laboratory scientist. Okay, so cytological methods, actually it's in histopath section. So um, specimen being submitted in the lab is being processed through a cytospinner, and they are being examined directly and evaluated by a pathologist. But for direct fluorescent antibody testing, it should not be used for genital tract, not routinely used for genital tract because it is labor intensive and requires experienced microscopies. Not to mention that it is expensive and you need to have a fluorescent dye, a fluorescent stain, and you need to have a fluorescent microscope. As compared to the cytologic methods, it is only about 75 to 85% sensitive, and cross-reactivity to other bacteria has also been reported as well. So a positive result here is this one. You'll be able to see fluorescence of elementary bodies. However, this needs further verification. So the picture that you have here is an example of a direct fluorescent assay. So cytologic method is much better because it is more practical and it is more sensitive as compared to the direct fluorescent antibody testing. Okay, so the surface antigens are used to prepare antisera conjugated to fluorescent dyes. So the specimen plus surface antigens. Surface antigens can be the MOMP plus the fluorescent dye. And you should be able to see the elementary bodies. Yung orange na nakikita ninyo rito, those are the reticulate bodies. But the small, bright specimen that you can see here, these are the elementary bodies. So this is the inclusion body from the ocular swab of a 7-day-old newborn. Wow. So this newborn has an inclusion conjunctivitis. So what are the advantages of the direct examination, direct microscopic examination? So it allows for immediate quality control of the specimen, revealing whether columnar epithelial cells are present or not. So you do not proceed with culture if you are not able to visualize the elementary bodies in direct microscopic examination. So remember, culturing chlamydia is actually more expensive because it would require you to have a live tissue culture. So you need to buy cell lines in order for us to culture chlamydia. So speaking of cell cultures, these are the possible cell lines that we can actually use. So the most popular cell line for chlamydia is McCoy cells. But we can also use HeLa cells. So HeLa cells are actually derived from the cell line of Henrietta Lacks, the black woman who died from the cancer of the cervix. And another possible cell line that we can use for the cultivation of chlamydia is the buffalo green monkey kidney cells. So cell lines are grown on cover slips in 1-drum shell vials on the surface of the multi-wall cell culture dishes containing the cell culture media. So we need to supply cycloheximide to suppress host cell protein synthesis. So after 48 to 72 hours, we stain the cell with fluorescent labeled monoclonal antibodies to detect if chlamydial inclusion is present. So if you're able to see, these particular cells, it means that you're able to isolate chlamydia in the cell culture. So it's kind of expensive and it requires also training because cell cultures are usually being contaminated by other bacteria if you're not really careful. Okay, so this is an example of the growth of the organisms in the laboratory that requires cell culture. So suitable cell lines. So aside from McCoy, Gila, and buffalo green monkey, we can also use the chick and embryo. So inclusions can be seen in infected cells. So here is an example when you stain the inclusion with iodine. So again, if it's seen inside the body, so these are inclusion buds. So these are another examples of inclusion bodies from Chlamydia trachomatis. This time, they have actually used McCoy cells as cell line for the cultivation of Chlamydia trachomatis. Okay, so immunoassays and NAAT. So these methods are non-cultured and this is the non-amplified. Okay, so we have the enzyme immunoassay. So here we use monoclonal or polyclonal antibodies against the chlamydial LPS. Okay, so it's not appropriate for urine or vaginal swab specimen. But for the nucleic acid hybridization and amplification assay, it's actually preferred because it has the ability to detect chlamydia and gonorrhea in one test. So it will allow us to detect and even differentiate chlamydia from gonorrhea. We need to do that because sometimes the signs and symptoms are actually confusing. So in order for us to differentiate it, we are using the NAATs or the nucleic acid hybridization and amplification assays. However, the most practical way of diagnosing chlamydia is through serological tests. Although, in general, it says here, serologic assays have little value. Though, serologic test is growing as a complementary diagnostic tool. So when you say complementary, it is not the the end all and be all of everything because clinicians usually correlate serological tests with the clinical history and clinical presentations of the patients. So one of the examples of serological tests is the microimmunofluorescence assay or the MIF. So this one is being used for detecting Chlamydia trachomatis antibodies. So microimmunofluorescence assay is characterized by the presence of organisms that glow in the dark. So this is positive, while these particular wells are actually negative. Microimmunofluorescence, the term micro is used because you need to visualize a specimen using a fluorescent microscope. So to detect ascending infections involving reproductive organs, uh, that may not otherwise be identified. So this one is very practical for that particular situations. So complement fixation will detect family reactive antibodies in systematic infections. So you know what, guys, if you pass bacteriology, of course everyone will pass by God's grace. So if you pass bacteriology, so the prerequisite subject of immunology and serology is bacteriology. So we will learn more about the serological test next sem. So in serology, we will be discussing enzyme immunoassay, complement fixation. And so, but as early as now, I'm introducing to you some of the serological techniques that are useful in detecting or helping the physicians diagnose bacterial or even viral infections. So again, chlamydia species can be detected by various serological methods. Okay, so perhaps you might be wondering what is 256, 32, or 128 all about. This refers to antibody titer. Antibody titer refers to the concentration of antibodies in serum. So next time, I will also be teaching you on how to determine antibody titer. Okay, so for Chlamydia trachomatis, as you can see, complement fixation test is very useful. Not only for Chlamydia trachomatis, but for Chlamydophila pneumoniae and Chlamydophila psittaci because they can detect an antibody titer of 205, 256. But for microimmunofluorescence, it is actually more sensitive because it can detect antibody titer as low as 32 or 16. And it can, and in order for you to detect if there is a fourfold rise, then you have to compare the acute and convalescent serum or sera. When you say AC, A stands for acute and C for convalescent. So acute means that you collect serum from the patient at the early onset of infection, and convalescent means that you collect serum again from the patient after several weeks. So that's the convalescent serum. And the comparison between the AC sera becomes significant if there is a fourfold rise. So for example, initially it is 4, and then after several weeks it becomes 16. So therefore there is a fourfold increase. Okay, so greater than 16 is considered to be as a significant fourfold increase. Okay, so that is possible for the MIF method. Now, IgG is being detected, and IgG usually is seen in convalescent serum, while IgM is usually seen in acute serum. Why? Because IgM is a primary response, IgG is the secondary response. Okay, so moving on, laboratories must clearly report and define test results. So which means that it's very important that we establish a form of communication with key personnel. Okay, particularly, um, we should be able to coordinate with OB/GYN and even the ED departments on which organisms are associated with clinical syndrome and then test accordingly. So report which tests were and were not performed for each patient profile so that we'll be able to provide the significance and interpretation of a positive results. Okay, so moving on. So we're done with the detection of Chlamydia trachomatis. Let's now discuss the detection of Chlamydophila pneumoniae. So Chlamydophila pneumoniae is formerly known as the TWAR, which stands for Taiwan Acute Respiratory Strain. But nowadays, Chlamydophila pneumoniae is now known as an important respiratory pathogen, especially nowadays amidst the COVID-19 pandemic, because clinicians have to be careful not to see, uh, uh, they have to be careful for the incidence of Chlamydia pneumoniae because it is a possible secondary bacterial infection among COVID-19 patients. Although there is only one, not server, but only one serovariety. Okay, there's only one serovariety. So 90% of infections are mild or asymptomatic. So patients with TWAR will usually have sinusitis, pharyngitis, acute respiratory disease, bronchitis, and pneumonia. Although it is also found in patients with otitis media with effusion, pneumonia with pleural effusion. So, ibig sabihin pleural effusion, in Tagalog, we say parang may tubig yung baga, and aseptic pharyngitis. So, it is implicated as a possible factor in asthma and cardiovascular disease. So clinical picture is among college age students are actually very prominent. Okay. and it is typically a biphasic clinical course. Pag sinabi natin biphasic, there are two phases in which phase 1 is characterized by prolonged sore throat and hoarseness of voice, followed by flu-like lower respiratory tract symptoms. And phase 2 is characterized by pneumonia, bronchitis without sinusitis or fever. So, of course, nowadays we do not want to experience these signs and symptoms because it might be mistaken for COVID-19. Now, infant pneumonia, unfortunately, is also possible. So the risk to an infant passing through an infected birth canal is high. So that's the reason why screening of mother before an infant is born is very important, especially for those who would be delivering the child normally. So the infant acquires the organisms during delivery with colonization in the upper respiratory tract, and eventually it will lead to lower respiratory tract infection. So that's so unfortunate for the newborn. Okay, so these are the summary key epidemiologic and clinical features of Chlamydophila pneumoniae infections. Okay. Um, for if there is almost no antibody detected before 5 years of age, okay, so that's one category. Antibody present in 50% of adults, that's another category. So attack rate is between the ages of 6 and 25, particularly if it's a college student, if the patient is a college student. Bakit kaya with emphasis on the college student? Siguro they are actually assuming that this particular student is in a dormitory and is exposed in a certain community with incidence of Chlamydophila pneumoniae. So that's another category. No incidence, no seasonal incidence. That's another epidemiologic category, and reinfection. So these are the 5 epidemiologic categories. So number 1 is self-explanatory because of the possibility of the infant's pneumonia or neonatal pneumonia. Okay, so these are the 5 categories of the epidemiologic patterns of Chlamydophila pneumoniae. Okay, so and then there are actually 4 population patterns for Chlamydophila pneumoniae. So we can actually group um, populations into 4 cohorts. Okay, so the first cohort are those people with pneumonia that would require hospitalization, and they are aged between 6 to 20 years. The second cohort of population are those college students with pharyngitis. Okay, the third cohort are retrospective undiagnosed outbreaks in young adult college students or military trainees. Okay, retrospective because at that because they were not diagnosed before. And then the fourth cohort are those population with serious pneumonia undiagnosed clinically present like Mycoplasma pneumoniae. So incidentally, Mycoplasma pneumoniae is the causative agent of primary atypical pneumonia. So this particular cohort would have a specific evaluation methods. So the common denominator among this evaluation method is the utilization of the MIF. And and the chances of very high antibodies are seen in cohort population number 1, 12%, followed by 9% antibody prevalence in cohort population number 2. So if a person is negative in MIF, it says here, then we should consider Mycoplasma pneumoniae. So Mycoplasma pneumoniae is the causative agent of primary atypical pneumonia, otherwise known as the walking pneumonia. So for the laboratory diagnosis of Chlamydophila pneumoniae, sputum is actually being is a specimen that we can submit to the laboratory, and then bronchoalveolar lavage. So this is an example of bronchoalveolar lavage. So we literally collect, we literally inject saline into the lungs of a person and eventually recollect the saline. So that's the lavage. Nasopharyngeal aspirates and throat washing and culture. So bronchoalveolar lavage is highly taxing and traumatic for patients. Culture, we can use the human lines and Hep2. Okay, serology, we're using the MIF or the ELISA or the enzyme linked immunosorbent assay. So this is how Chlamydophila pneumoniae is detected using the fluorescent antibody test. So you can see that the organism also glows in the dark using the fluorescent microscope. Okay, so let's talk about the last organisms for tonight. And this is the Chlamydophila psittaci. Okay, so seen among the cetacean birds. So the avian species can cause psittacosis among cetacean birds. So that's the reason why the infection is known as the ornithosis or the parrot fever. So as you can see in the picture here, the birds would actually the bacteria in the bird can actually be transmitted to humans. Okay, because of our close contact with these birds, because these birds are also being used as pets by humans. So aside from humans, cats can also be infected, no? So Chlamydia psittaci causes feline conjunctivitis, rhinitis, and respiratory infection. Okay? So in guinea pig, Chlamydia psittaci causes conjunctivitis as well. Okay. Human infection of psittacosis is also a possibility. So it is through inhalation of the bacteria following contact with poultry. So which means that we would be of greater risk because of this particular mode of transmission. So person to person spread seems unlikely. So it's more of bird to person spread. So in humans, symptoms vary from none, which means that sometimes you are asymptomatic, to severe pneumonia. So it's kind of extreme. So characterized by fever, chills, muscle aches, severe headaches, and occasionally gastrointestinal upset. So we make diagnosis of parrot fever based on clinical history. So we probe if a person is exposed to birds or not. So there should be a four-fold rise in antibody to chlamydial LPS antigen. So again, when you say fold-fold rise, we compare the AC sera, the acute and convalescent sera. So misdiagnosis can be a problem due to similar symptoms, similarities of symptoms, and bird exposure. So usually yung avian flu, so it can be mistaken for avian flu. So it is important for healthcare providers to know the tests that are most appropriate for the situation. So for Chlamydophila psittaci, culture is not recommended, although diagnostic, but it is difficult and dangerous. So a single antibody titer of greater than 1:32 is suggestive of acute illness if the person is asymptomatic, particularly whenever there is an outbreak of psittacosis. So titer has become weak or absent if the person has already started antibiotic therapy. So if IgG and IgM specific to both Chlamydia pneumonia, Chlamydophila pneumoniae, and Chlamydophila psittaci are not detected by MIF, so a four-fold titer described above is detected. So if we cannot detect IgG and IgM, but there is an increase in the four-fold titer, then we should suspect Chlamydia psittaci, which means that the doctor would really have to probe if a person has been exposed to birds or not. Because it wouldn't make sense if a person is infected with Chlamydia psittaci if he or she is not exposed to bird, because human to human transmission is not possible. So a good patient history is therefore very helpful. Okay, so that ends the first part of this particular module.